Greetings to our instructor, Miss Elaine Joy Torre. This is our oral report about Experiment 5 carbohydrates. So, what are carbohydrates? Carbohydrates are one of the four biomolecules consisting of carbon, hydrogen, and oxygen atoms. Additionally, they are considered as part of the basic food groups, and usually they serve as a principal source of energy, but they can also help control glucose and insulin metabolism, participate in cholesterol and triglyceride metabolism, and help with fermentation. So, what are carbohydrates are made of? The building blocks of all carbohydrates are simple sugars called monosaccharides. So, a monosaccharide can be a polyhydroxyaldehyde, or you can call it aldose, or a polyhydroxyketone, or ketose. As you can see in the example, sucrose. So, sucrose have monomers of glucose and fructose. So glucose is an aldose sugar while fructose is a ketose sugar. Carbohydrates have chiral or asymmetric carbon centers with four different groups attached, giving rise to stereoisomerism. So stereoisomers are optical isomers that tend to form molecules that are mirror images of each other. Also, carbohydrates can form multiple hydrogen bonds due to the presence of different functional groups in their structure. In experiment 5, tests for carbohydrates, so we only conducted qualitative tests which are used to determine the presence or absence of a compound in a given solution, and in this case, carbohydrates. Although qualitative analysis of carbohydrates may encounter difficulties due to the chemical and structural similarity of the samples, like their stereoisomerism, but there are several rapid tests that are already available, such as tests based on specific color reactions that can be utilized to determine the presence or absence of sugars in a given sample. So, in experiment 5 includes 6 different tests to assess the different properties of carbohydrates. So, specific reagents are involved and introduced to each samples to find results. One of the tests or qualitative tests in carbohydrates is Molesch test. So Molesch test is the most common test for carbohydrates. So it is the general test for all carbohydrates, whether a substance has a presence of carbohydrates, um, it will be positive in Molesch's test. Benedict's test and Felling's test are specifically tests for detecting reducing sugars. So reducing sugars are mostly simple sugars, whereas Barfoed's test is used for detecting presence of monosaccharides in given samples. So if the sample of substance have a more complex sugars like starch, or even disaccharides like sucrose or maltose, it will be negative in Barfoed's test. While in Sullivan's test, it is a test used for distinguishing between aldose and ketose sugars, while the iodine test is the qualitative test for determining if the sample is containing starch or complex sugars or polysaccharides. For the experiment, 12 samples are used for the different tests, which are fructose, sucrose, ribose, maltose, which are provided in the laboratory, and the brown sugar, starch, rice with saliva, boiled rice with saliva, bread without saliva, bread with saliva, and boiled bread with saliva are prepared by the students. Along with the samples, different chemical reagents are provided in the laboratory, such as the Benedict solution, Barfoed solution, Slavonow's reagent, iodine solution, Molesch reagent, and Fehling solution A and B, which are obviously utilized in the specific test for carbohydrates. So the concentrated hydrochloric acid and diluted sodium hydroxides are also provided. Test tubes, beakers, droppers, and other equipment are also used. During some experiments or tests, a laboratory bath was used to heat the samples in the lab. So tests such as Benedict's test, Barfoed's test, Slivanon's test, and Felling's test are used this equipment to successfully get the results.
In this discussion, I will be discussing the use of qualitative test for carbohydrates. So first, Benedict's test. This test is used to detect the presence of reducing sugars. But first, what are the reducing sugars? Reducing sugars are all carbohydrates that contain either ketone group or aldehyde group. So, if a sample contains either of that, it will show a positive result with orange solution. Whereas Molish test is the test used to detect the presence of carbohydrates in a given sample. This test involves the use of Molish reagent. So, if the carbohydrates are present in a given sample, it will show a positive result with peach and purple solution. Felling's test. This test is used to differentiate between reducing and non-reducing sugars. So, it can also be used to distinguish ketone group carbohydrates and water-soluble carbohydrates. So, in this test, if the given sample show the positive result, it will show or form a red solution, indicating the presence of reducing sugars. However, the Barfoed's test is used for detecting the presence of monosaccharides in a given sample. So, if the given sample show the positive result, it will show the formation of red copper oxide precipitate. Selimanov's test. This test is used to differentiate between sugars that are ketone containing and sugars that are aldehyde containing. If the given sample contain either ketone or aldehyde group, it will show a positive result with the variant color of red. However, the iodine's test is used for detecting a given sample if it is starch containing or non-containing. If the sample contains starch, it will show a positive result with dark purple solution. So if it's not, it will show a light color of brown solution. Based on the result from Benedict's test, again, test for reducing sugars, most of the samples showed positive results, which are fructose, sucrose, maltose, ribose, brown sugar, rice with saliva boiled, bread without saliva, bread with saliva, boiled bread with saliva. However, the samples that showed negative result are starch and rice without saliva. In Molish test, the samples showed only negative results are rice with saliva boiled, bread without saliva, and bread with saliva boiled. Based on the result from Fenning's test and also a test for reducing sugars, most of the samples showed positive results. And as you can see, the results from Fenning's test is different from the results of Benedict's test. However, the results from Barfoed's test, a test for monosaccharides, half of the samples showed positive results and the other half showed negative results. Based on the result from Siliwanov's test, a test for distinguishing aldoses and ketoses, 6 samples showed positive results and 6 samples showed negative results. Last, the result from iodine's test and test for starch containing, the samples showed negative results are fructose, sucrose, maltose, ribose, brown sugars, and the sample supposed to be negative, rice with saliva and bread with saliva.
To summarize the conclusion and recommendations, this discusses the quantitative analysis of carbohydrates through chemical reactions with specific reagents. The presence of carbohydrates is indicated by the development of a distinct color. Monosaccharides such as glucose are expected when rice and bread mix with saliva due to the catalytic action of salivary amylase. However, Molish tests yielded negative results for rice and bread samples due to the experimental errors. In tests for reducing sugars like Benedict's and Fellings, certain samples produce unexpected positive results, indicating errors in the experiment. Barfoed's test for monosaccharides and Seliwanov's test for keto sugars also show discrepancies in results. Iodine tests for polysaccharides exhibited unexpected color changes in rice and bread samples with saliva, suggesting incomplete breakdown of starch by salivary amylase. This concludes that false positive outcomes, especially in rice and bread samples, highlights the need for better sample preparation to ensure accurate results. It emphasizes the sensitivity of carbohydrate tests, urging contamination-free samples and precise handling for reliable outcomes. Additionally, it suggests exploring alternative methods for complete starch breakdown to glucose, such as using pancreatic amylase in the small intestines.
So that wraps up our oral report. Thank you and have a good day.